right, everyone. Good morning. Thank you for joining us. We're going to go ahead and get started. I see about 35 people in here, so we can go ahead and get the presentation going. So thank you for taking time to join us today for the June IMEDS community call. This is Nicole Spear from the Reagan-Udall Foundation. We're going to try to keep the phone off of mute today so you guys can kind of jump in where you need to or want to. So we'll keep that going for as long as we can, but if we start to get background noise, I'll just take a moment to pause and put everybody on mute and then unmute the presenters. So today we have with us Dr. Rima Izem. She's the lead mathematical statistician at CDER's Office of Biostatistics. She's going to discuss with us some of her working group learnings and experiences in the IMEDS Research Laboratory. So the slides being used today were sent out to you yesterday. If anybody needs a copy of them, just let me know and I'll email them over. You should see them in front of you as well. And then we will be posting them to the IMEDS website as usual in another day or so. So Dr. Izem, first, thank you so much for taking time to be with us today. And with that, I will turn it over to you.
Thank you, Nicole, and thank you, Susan, for inviting me to speak today. So as my title suggests, I'll be sharing our working group experience using IMEDS to learn more about claims data. This is my disclaimer. The views represented here are my own and do not represent necessarily the views or the policies of the Food and Drug Administration. This is a second disclaimer. So Susan had to twist my arm a bit to give this talk and convince me that my perspective may be of interest to other users in the lab. And the reason for that is not that the problem was with talking or sharing knowledge, but I didn't feel that we were quite ready to do that. But my presentation is really focused on starting the conversation rather than telling you what we have done, because we're still working on some of our goals. And I hope that this perspective will start a dialogue with other users of the lab, whether they're thinking about using or they're new users like ourselves. If you're thinking about using the lab for teaching, then some of our experiences may be of interest to you. And on the other hand, if you're a tool developer, then maybe our perspective will be interesting to you if you're developing some content. So with that in mind, with that audience in mind, I wanted to describe who we are and what we do and why access to the IMED lab was so important for us, and then what we have done so far and what we plan to do. And I wanted to get an idea of whether I was sort of targeting this to the right audience. And if you log off now, I won't take it, I won't blame you for this, but I prepared some polling questions just to give me an idea of who you are. So let me see whether I can. So each question will ask you something about who you are, and I think apparently 100% of the participants are in government. Is it possible to redo the poll, or should we move on to the next question?
Give us a moment. If you want to move on to the next question, we'll work on that. Sorry about that. They must have answered.
Okay. All right. So I think we've had 26 responses so far. Did everybody who wanted to answer the poll answer? So that's a different composition than I was expecting. But I'm glad for that. So this talk may be relevant for especially those prospective users and the new users as well. That's great. Should we move on to the next question, or are some people still trying to answer the poll? Okay. All right, one question. So your familiarity with the OMAP experiment is best described as no familiarity, some familiarity, or very familiar, or part of the research team member. Okay. All right, most of you are familiar with the OMAP experiment. That's great. And then finally, just drug safety and how it relates to your work. I evaluate drug safety, that's me. Does that relate to your work, yes or no? All right. Oops, I closed it maybe a little bit too early. I almost had perfect compliance. So yes, most of you do work somewhat related to drug safety. Thank you so much for answering these polls. This is the first time that I was using the tools, and really it does help a lot. So let me go back to the presentation now and continue. That will help tune how much I say. And as Nicole said, feel free to interrupt me, and if for some reason all of you are on mute, there is a way to raise your hand, and I'll try to keep track of the participant tag to see whether you raise your hand to ask a question. All right. So who are we in the IMEDS working group? We're all volunteers. We all do that, meaning as a hobby. It's outside of, it's in addition to our work, but it's not our primary work, which is mostly regulatory review. Our members are from both the Division of Biometrics 7 in the Office of Biostatistics and the Center of Drug Evaluation and Research, and also epidemiologists from the Division of Epidemiology 1. So it is multidisciplinary across 2 different offices. I started this working group, and I got it going. And last year, Yo-Chin Zhao, or early this year, Yo-Chin Zhao agreed to take over the leadership, but I'm still very much involved in the working group. But she's making great strides actually on accessing the lab. So if you have questions at the end or during the talk, I may be asking her to help me answer the questions. We have advisors as well, people who are making sure of what we're doing sort of fits with the FDA mission, and also that we stay on track. So in the who we are, most of us are, as I said, in the Office of Biostatistics, and this is the organization chart of the whole Office of Biostatistics. I'm told that we are the largest concentration of biostatisticians in the world under one office. Our office has over 180 statisticians. Most of us in the Division of Biometric 1 to Biometric 5 are divided by therapeutic area. Division of Biometric 6 is across different therapeutic area, and we sit in Division of Biometric 7. So Biometric 8, which is our newest division, is for generic drugs. So in Biometric 7, we have 4 different teams. By and large, the first 2 teams, Safety 1 and Safety 2, look at safety in the pre-market setting. And then by and large, our work in Safety Team 3 and 4 are looking at safety in the post-market setting, mostly with observational data. So what does it mean to look at safety in the drug development? This is a very simplified cartoon of the drug development process, starting with basic research and ending in post-market assessment. Safety is part of our mission at the FDA of protecting and promoting the public health, and we look at it in every stage of the development process. First, before a drug is used in humans, we make sure that the basic research support that it is safe enough to be used in humans. And then obviously before the drug is put on the market, we make sure that the benefit outweighs the risk. And then after a drug is marketed, we continue with the safety surveillance. So before we get to safety surveillance, we have a pretty good idea of what the drug safety profile is, sometimes from the marketing application. So the division of biometrics 7 straddles, as I said, the pre-market and the post-market side. And that's quite unique among the different, our other colleagues who work in the Office of Biostatistics. And what I will focus on here is mostly on observational studies, which is in the post-market assessment. So what do we actually do? We're regulatory scientists. This means that we get excited a lot about statistical methods that are used in observational studies because they affect our work. But also we get excited about the regulatory side, which is how do you communicate the findings from these post-market assessments to the public, to health scientists, to physicians and patients. So most of our work is to review other people's studies, whether there are published studies or industry studies from meta-analyses, and we review the results and we think about how to integrate that or whether they have implications on the regulatory side. One thing that is unique about our team in the Office of Biostatistics is that we get also to conduct our own studies using resources such as Sentinel or our federal data partners, that is Medicare, Department of Defense, or the VA. So those are all sources of observational studies that we can use to answer safety questions of interest. And so anytime there is a safety question of interest that require a look into observational studies, we are part of the team with epidemiologists to write the protocol, write the specification, write the statistical analysis plan, and then review the results and write them in a report or a published paper. We also have access internally at some observational data at the subject level, not just at the summary level, and that's, as an example, it's the CPRD. So the reason I bolded that is that by and large, most of our work is not actually accessing subject level information, which is a big difference between what we do in post-market and what we do in pre-market in our office. In our office, when you review pre-market applications, you have access to clinical data, to the subject level information of the findings. However, when you do post-market data like we do in our team, often you don't have access to the observational studies that were used. And that has big implications about what we request from others to show us in their reports, and then also the level of details that we need in the reports to explain the results. And then it will also give you an idea of how much experience we have doing observational studies within the FDA using subject level data. And this says that really we don't have as much exposure to subject level data as maybe I would want, for sure. So that's the biggest motivation to access the IMEDS lab. And when this was advertised at the OMAP conference in November 2013, I was very excited because that was when I moved to doing post-market reviews, because it gave us like an unprecedented access to claims data to learn about the data collection and the data quality, to look at the strengths and limitation of the different designs and methods, again, keeping in mind that we have sometimes to review protocols and statistical analysis plans, so it's important to understand those strengths and limitations. Also, what are the diagnostics for different statistical methods that are most useful to have in a study report to sort of assess the strength and limitation of them statistical methods. Although most of our motivation for accessing the lab is actually learning about claims data, eventually we hope that down the road, under another sort of mandate, we can, if we do identify gaps in knowledge, we could also use the lab to develop the new statistical methods. But that last bullet is not our primary purpose right now. It's more looking into the future after we're done with just some sort of exploratory analysis and learning, we could use that as a resource in the future. So any questions of who we are or what we do so far before I move on to what we're doing or what we did in the IMEDS lab? Okay. This slide here summarizes our timeline from when we started the IMEDS working group to what we're doing right now. And then the next few slides will actually go into a little bit more details in each bullet point. So the first thing that we had to do is access the IMEDS lab, and that was relatively quickly thanks to the IMEDS group. We wrote a proposal, we submitted the proposal, we had a phone call to sort of make sure that everyone understood the scope, that it was mostly for learning purposes, and we signed the agreement to join the lab, and we got usernames and passwords for everybody in the working group. So in a couple of months, all of that was set up, and so the agreement was reviewed by a lawyer internally, and it was all agreed upon. So that was fairly quick. All of us in the working group had different levels of knowledge of what the OMAP experiment was and what the goals and the methods were. And in order to put everybody in the same level, so to speak, we reviewed a lot of the videos, and also we discussed some papers that made us more familiar with the methods and the findings. Finally, we picked an example to design and test, and then since it's been almost a year now that we actually have started going into the lab and modifying the code for our example. So through that, we had a better understanding now of the parameters in the OMAP experiments, and then also what are some of the codes that they used. So let me walk you through some of the more details. Oh, and then finally, as I said, the primary purpose here is teaching. So we have been keeping record of our minutes and of all the documents that are useful for others in a shared folder. And I'll have a screenshot to show you that later. This is my very blurry picture of our one of our labs in the Office of Biostatistics. And we have one machine that's on the scientific network that we call Tukey. So that's what we've been using primarily to access the scientific lab. I started this presentation saying that I was hoping that this presentation would be the start of a dialogue. Well, I can tell you that while I was preparing for this presentation, Susan Gruber had put me in touch with other prospective users at FDA that are in different offices. In the Office of Biologics and CDER. And I already found out just from talking to them yesterday that there are other resources at the FDA for us to access the scientific network. So for those of you, I guess, who may be wondering, like, how come does everybody at FDA have access to only one machine to do their work? No, that's not what we're talking about here. Each one of us have plenty of access to many computers. However, to access the lab, you're working in the IMEDS cloud. You're not downloading data onto your own computer to do the work. And so this network access was a little bit of a concern for us. We wanted to make sure that we could access the IMEDS lab without putting the FDA network, regulatory network, without putting a threat on that network, on that connection. So for that reason, we're using the scientific network at FDA rather than our regulatory network at FDA to access the IMEDS lab. So we found that whatever method you choose to use to access the IMEDS lab, that it is helpful that the machine that you use, that you have some limited administrative privileges, because there is a client install that is updated from time to time in the IMEDS lab, and that you do have to update in order to use the lab. So to learn again about OMAP, I'm putting here the links of the different conferences. this was just also a reminder to say that the 2012 conferences for some reason were on YouTube, whereas the 2013 conferences were, I think, on another platform. And so if you watch a 2012 video, you can actually go back and forth. But then 2013, once you start the video, you can just stop it, I think. And that's important because sometimes you hear something and then you want to stop and discuss and then go back and make sure you understood it correctly. And we found that it's much easier to do it with the YouTube videos rather than the 2013 conferences, which are in the different platforms. The other thing that we noted as well is our primary, the biggest motivation for us for starting this working group was the OMAP 2013 conference that made a very strong case that the data set was available, it could be used by everyone, and we took that really to heart. And it is true. The data is available, it's easily accessible, and everybody can use it. They even had, at the conference, a sample code in SQL and R to get everyone started. And that was our starting point as well. However, we have noticed that the code itself is not available on the OMAP website, at least last time I checked. And in the future, if the IMEDS lab can have it easily accessible for new users, I think that would be a great resource. For us, there were a few of us who attended the conference, so they could share the code internally with those in the IMEDS working group. So the code that we've been modifying specifically is one of the ones that was used as an example of warfarin and GI bleeding. That we changed it for a different drug and for a different outcome, but that was our starting code in SQL. So okay. So we put all our meeting minutes and material and different codes and all of that. We plan to use we are using the SharePoint at FDA. And then I wanted to highlight that in our lab material, we have a step-by-step instructions on how to access the IMED lab in Linux. This was developed by Mark, and we also made some we added some other screenshots that are specific to our network and how to use it. So again, those are resources that we're happy to share with everyone. We don't want to reinvent the wheel for everybody who's a new user. We wanted to modify the code for a specific example, and we didn't want to pick a new example. This is a learning exercise. We wanted to put an example that was tried and true. So one of these examples that was recommended to us was the ACE inhibitor and angioedema. So it is thought of as a fairly robust example where the association has been shown in different databases, including the Mini-Sentinel database, and I'm showing the references here for the Mini-Sentinel database. And the plan for us was to take this example and not so much worry about the scientific question or the safety question, but use it as an example to test out different methods, test out different designs. And I'm not saying that case control, effect control are better designs for this question. Actually, cohort is probably the most appropriate, but we just wanted to try different designs because we wanted to build we wanted to look at the code for these different designs and learn how to use them. So the advantage of working with something that's tried and true is that probably if there is a mistake, if some of the results are funny, then you will know for sure that it comes from your code and it doesn't come from the data or maybe your drug outcome association. So one advantage of this example is that it was part of the OMAP experiment, at least the 2011 experiment. So you see here I highlight for angioedema, both the ACE inhibitors and the beta blockers were considered as one positive pair and the other one was a negative pair. So that's for our example. So what do you do when you get a code? Well, you look at the variables and you spend a lot of time poring over the documentation. So this is the cover page that you're probably, most of that are new users are familiar with from the Common Data Model version 5, the manual. So the things that were pretty, for us, at least for me, easy to understand is person characteristics, drug exposure, and condition occurrence, and those are variables that we've had to use. the things that are still trying to understand better are the standardized vocabulary and the standardized derived elements. Now, if you went to the OMAP presentation, or if you watched the video from the 2013, there was a whole presentation devoted to the standardized vocabulary, understanding, the relationship, the parents and children. At the time, it seemed very clear until I got into it, and then things were started to get a little bit less clear or more nuanced. And certainly, I would appreciate some help from the user community to understand these a little bit better. So let me tell you what I mean by that. So in our test case for ACE and angioedema, we knew that we had to change the code in terms of determining what are the codes for the exposure, what are the codes for the outcome, and what are the inclusion-exclusion criteria. How can we change the inclusion-exclusion criteria to adapt it to our cohort specification? So in our cohort, the main drug of interest is the ACE inhibitor. The comparators are the beta blockers. And to determine the treatment periods, the protocol, at least the Sentinel protocol, has used gaps of 15 days in treatment to define a continuous treatment episode. And then it included subjects that are 18 years or older, that for whom in the look-back period there was no use of ACE or beta blockers. This is a new user design. There was no angioedema, so this is also we wanted to look at new cases of angioedema, and then no initiation of more than one drug at index date. And then the outcome was angioedema, and the paper, the Sentinel paper, had the ICD-9 code for that. So in the common data model, we found the corresponding variables that we had to change, and this is what we thought would work: the concept ID, the ERA start date, the ERA end date. And our understanding is that some of these derived variables had a default for defining a use period that is of 30 days. But that's something that, if some of you are more familiar with the code and can comment, that would be useful for us to know, because that's our current understanding. Finally, you can use some variables in the vocabulary for subject characteristics like year of birth. You need to define what a first user is and what the condition concept is, and then some cohort variables. And then the outcome was already defined as a health outcome of interest in the OMAP vocabulary. So it was very easy to find also the outcome, an outcome date derived variable. So the reason I say that some derived variables are not as intuitive as I thought they would be is some of what we call derived variables are actually user-derived specification. The user gets to specify what's the cohort start date, what's the cohort end date, and what is a cohort, and what's the first user, and etc. However, some so-called derived variables are not necessarily user-specified. There was actually some background work already done from start date to define a drug ERA, for instance. And so this means that this was already derived. The user doesn't have to rederive it, rederive the definition of what the drug ERA start date. At least that's my understanding. The other thing here that you'll notice, I guess if you're a more experienced drug user, notice here that my exposure, whether it's the main test drugs, the ACE inhibitor, or the beta blocker, are actually not just one drug. They're a class of drugs. So the variables that I'm listing here from the common data model are actually drug-by-drug variable, not necessarily associated with a drug class. So, and I'll go back to that a little bit later in my talk. So what are the challenges that we faced? Before I get to this slide, let me summarize sort of some of the challenges that we've discussed so far. So access was not a big challenge, but it was somewhat of a challenge to just determine how you can access the network securely. Time was a challenge from the beginning. As I said, this is not our primary work. It is a hobby. Sometimes the only time that we had to work on the code was 1 hour every 2 weeks to sort of, so you have time to re-immerse yourself into this and then start working on it. Language. SQL is not our primary language by far for most of us statisticians at least, and we've had a lot of help from our epidemiologist colleagues for programming. And some of us in the group, like Yochin, is much more experienced than others. But SQL is a foreign language for a lot of us, so that's the third challenge. So here is also a challenge about the data structure that I wanted to get to in the next 2 slides. As statisticians, we're very, 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 at least statisticians in the Office of Biostatistics, we're very comfortable with data matrices. You give us a data matrix, whether it's a matrix by where the rows are subjects and the columns are variables, or whether it's a long skinny matrix that is, say, in the CDER standard. You give us a matrix, and we can run our suite of statistical analysis to determine both baseline characteristics, to run diagnostics, and to also look at outcome models. We're very familiar with that data structure, and we have all the tools to do that. In the IMEDS lab, there is a bit of an extra step that you have to do. So the database is not actually, I mean, each part may be a matrix in itself, but the whole is a linked databases, and what's linking them is this common data model. So that's a slightly different architecture for the data than we're used to. So that means that sometimes, and some people, some of the code that was illustrated, at least some of the test code that we have, went from the data structure itself to write customized code to do the statistical analysis and all the diagnostics. But some other sample code was going through the steps that we may be more familiar with, at least as statisticians, is taking this linked database to first get to a data matrix and then run traditional stat software to do the statistical analysis. So I don't know whether you'll see my mouse here, but this arrow here from the linked database to the analysis matrix is where we are now. And what we want is to do this, the statistical analysis, because that's we're statisticians, that's what we're most interested in. And we know the traditional stat software, we don't want to reinvent the wheel. However, in this sort of, it's not just a buzzword, it is big data, and it is rare outcomes, rare safety outcomes. So we understand that maybe it's only a matter of time before we have to do this arrow. Maybe we do have to think about writing customized code from this data structure to the statistical analysis. Maybe we have to write the code to do it efficiently and faster. So that's sort of my my spiel about the structure of the data, and that's how this has been a bit of a challenge. Okay. So so where we are now, this is only the start. We have realized that our example was not perfect because it wasn't one drug, one comparator, one outcome. It was one class of drug against one class of drug and one outcome. So because of that, Mark is helping us with developing some customized code, so to get us through the hurdle again of like going from this linked database to something that's more like a matrix format that we can, or matrix idea that we can use in our further analyses. And then we realized also that the drug gap, as we understand it, the default was set to 30 days. And we would like to understand better how we can modify this, because we would be interested in doing sensitivity analysis by varying the drug gap. The next step after we go through this hurdle is actually do the analysis, do the exciting part. Try different survival methods with propensity score adjustment, explore the code for propensity score diagnostics. In terms of other study design, we wanted to test the code for case control, extracting a self-control group from which we could run different types of analyses. So that's what we're hoping to do in the future. So that's it for my talk. I'm sure I would like to thank everybody in the IMEDS working group, including our advisors, Susan for inviting me and for all her help and support. Mark and Paul for getting us set up, and then all the countless OMAP of people who we've been in touch with every time we run into some issues. Thank you.
Thank you so much, Rima. So we've got about 20 minutes, and I'm happy to open it up for a question-answer period. If you can just announce yourself and let us know who you are before speaking, that would be great.
Well, this is Rich Place at the University of Pittsburgh. Thank you so much for taking the time to walk through everything that you've done. It's really informative for a person like myself who's interested in building tools that will support your drug safety research that you're doing. And I also have a hat where I do drug safety research in the nursing home setting. So I'm learning a lot from watching how you guys are walking through things as well. I wondered if you could elaborate a bit more on the issues you're having with moving the data from the common data model into a structure that the statistician could use. In my own experience at a clinic in an academic environment, basically you kind of need a couple people on your team to do this kind of work. You need an analyst who really understands SQL, who can write these rather difficult queries that involve some inner joins and sometimes pivots and various things, to pull out basically this operational data set that's more of a simple matrix format that could be imported into the statistical programming. It sounds like you guys as statisticians tried to take on more of that analyst role that I would think would need to be on the team. Is that kind of what ended up happening in your project?
Yeah, I think you're right, and it's fair. Like we didn't, well, we were trying to pick a simple example so that we can reuse code that was already developed, so we didn't have to. It's not that we don't think that part is important. It's more that we wanted to bypass that to get to where we wanted. But then we realized that by the nature of the example that we took, it wasn't as easy as we thought to reuse the code that that was used as an example to sort of modify it and change it a little bit and then go to the next step. So, but I totally agree with you, and that's what we, in our FDA-led research, we do work very closely with programmers to handle the complexity of the claims data. Like when we have partners with Medicare, we have Acumen programmers who deal with the complexity of the data so we can focus on the statistical programming part. But in this case, accessing the IMEDS lab was just a way for us to play directly with subject level data, and we wanted to make the first steps, like we didn't pick a new safety question, we picked a known example. We were reusing someone else's code so that we didn't have to make big modification to get us through the hurdle of doing the interesting part. But yeah, I agree with you. If this was used for research purposes or for investigating a safety question, we would have to work with programmers in SQL to do it right. Does that answer your question?
Yeah, I think it's just good discussion to have. You have up on the slide that it's sort of linked databases with a common data model. Are you referring to that in the IMEDS lab you have let's say the 3 Truven databases? Is that what you mean?
Yes. Sorry, I didn't mean that the 3 databases were linked. I meant that you have standardized clinical data, standardized vocabulary, and standardized health economics, and all the standardized health system. All of that is linked to each other. That's what I meant. I didn't mean that the 3 databases like MarketScan and GE and Medicare are linked. That's not what I meant.
Okay. So there's a lot of echo on the line, so I'm trying to kind of push through it. Hopefully it's not too distracting to other folks. I think the way that we in informatics would call this common data model is sort of a hybrid between a relational data model and an entity attribute value model. And like the slide that you had prior to this, or maybe it was two previous, where you sort of, could you go back, I think, one slide perhaps on the, this one right here. That's sort of representative of an entity attribute value model that's where the reason is that there could be any number of labs or conditions or drugs in the world that you want to analyze. And so the common data model has to be able to support all of those things, and it depends heavily on a terminology. That's what the standard vocabulary provides, is the variable names and so forth. And so, yeah, I think in my experience, there are other approaches to building sort of a research-oriented database. Like, for example, Picori, in their network, Pittsburgh, I'm not involved with the project, but we have another group that is. They decided to work with the I2B2 sort of approach, and it has even less relational structure than this. So you can imagine that actually, doing the queries becomes even more challenging when you don't have quite, even like a condition table, for example. I'm not saying that they don't have a condition table. I'm just saying that my experience from looking at it and talking to the folks is that it's even less structured than this. So it's really hard to find that balance when you're building this sort of a data model to support anything. And I think they've struck that balance. But what I hear you saying is from an analytic perspective, we still have a long ways to go to make the actual development of the actual development of the operational data set that can be used in the statistical analysis easy to sort of generate. And I think that's the direction that we're trying to go in the Odyssey collaboration. So if you look at some of the new Odyssey tools that are being developed, like there's a cohort explorer where it's basically a web-based interface for it interacts with the common data on the back end, but what you're presented with as a user is some drop-down lists and so forth that you can sort of build your cohort from. And I think that as that platform evolves, a lot of the problems that you experience will kind of go away, and the biostatistical folks will have a lot easier time designing the cohort from like a web-based interface and then getting the operational data set sort of exported. That's what I suspect is going to happen, as long as everybody stays involved and like folks like you continue to provide feedback, because it really does help drive the use cases on that project.
Yeah, thanks for that input. Yeah, we do go to the Odyssey website, and we did notice that there is a new R library in March, Get Cohort, which sounds very promising. We considered using it actually here, but that particular library was constructed for one drug, one comparator, one outcome. So it's not perfect for our example. So then part of me thinks, well, should we just switch the example to maybe instead of the whole class of ACE inhibitors and the whole class of beta blockers and this outcome to just the most common of drugs in each class, so that we can actually use these tools and take us through this hurdle? Or should we just try to push through and get to write that part of the code so that we can actually get to the interesting part? So that's a discussion that we're having within the internal group, and different people have different opinions. So that's where we are.
Have you tried to post an issue on the Odyssey community forum on that topic? Because I've seen some success when people come with specific use cases like you're having, where the other groups that maybe have developers dedicated to moving that forward might be able to help you to support that case.
Mark in the IMEDS lab is helping us, at least in IMEDS. And yes, we've had, you're the second person who's suggesting that we post this in the Odyssey. I don't know, Susan was asking me yesterday whether I had posted these questions in the IMEDS lab user group. And to be honest, at least for me, it's intimidating when you haven't seen questions that were asked by other users that are simple questions, to ask a simple question as well. So, but yes, if we have to be the first people to ask a question, then maybe we should do that.
Yeah, I would suggest not to be intimidated, because someone's got to start the conversation. But I do know exactly what you're talking about. The other question is, what is the statistical language that you guys are primarily using?
I can't speak for the whole group, but by and large, most of us use SAS and R.
Okay. That's great, because I know that R is one of the ones that's being strongly supported moving forward, so you'll see a lot more development of methods, I think, using R. Okay. I'm hoping to hear other people take over the conversation so I don't hog it.
No, thank you very much for all your questions. That's very helpful.
This is Patty St. Clair from the Schaefer Center at USC. And I'm just wondering, I guess, is there any kind of drug classification, therapeutic drug classification, that could be linked to the OMAP tables to do something like pull all the drugs from a particular drug class. And I recognize that that's a moving target, but it would be very helpful because a lot of people use drug classifications rather than specific NDCs in their research.
Yes, you're correct. I'm sorry. I apologize for not having mentioned that. I meant to. If you go back to the slide where I had the standardized vocabulary, actually each drug, we found the drug code and also the concept ID for the drug class. So that wasn't an issue. All these relationships are already built in the common data model. So there's been a lot of work already done to establish that. Something that was a little bit not very intuitive for us was that the link between the drug and the drug class wasn't always sort of parent-children. Sometimes there were 2 steps before you get to the right ancestor. So for instance, one drug, their immediate ancestor would be the drug class, but for other drugs in the same drug class, it would be 2 steps before you get to the drug ancestor that's the drug class. So that was one thing that we noticed that originally what was a little bit counterintuitive. And then the relationship between drugs and the drug class were actually already built. However, the derived element for drug era,
right.
that was what is constructed on a drug by drug basis. So it's a derived element because you have to understand what is the drug and how long has it been taken, how much gap there is between one instance of taking the drug and the other instance, and there's already sort of a lot of background work that was done. And this was done, as far as we understand it, on a drug-by-drug level rather than on a drug class level.
So would it
Does
be
that make
useful?
sense?
I think it would be useful to think about add-ons to the drug era table to have a drug class era table. I mean, for things like medication adherence measures and such, that would be very useful. And it seems like you could construct that easily based on drug class rather than particular drug. I don't know that would be a benefit at some point maybe in the future, or if someone else develops it, code to do that.
Yeah, I agree. Mm-hmm.
We do that all the time, but we haven't been doing it off of the common data model. So.
I think you could put in the drug era table, I think you could put the concept ID for the class instead of the individual drug. I'm not suggesting that. I'm not saying that's the greatest idea, but I think because the class is available as a concept ID, the same principle might apply if that's what you needed to do for research. It depends on how the data gets loaded. So the decisions get made through this process called extraction, translation, and load. And Mark Cater's got these ETL documents for all of the data that's been loaded in there. And they just made the decision, I think by default it would be at that drug individual, like clinical drug level. But I think it could be decided to append also drug classes if it was needed for that purpose, rather than, and then you wouldn't have to change anything as far as the schema goes. You just have to know that those drug class IDs are in there. But I'm not entirely sure if that's true.
Well, and also I think when you're deriving the drug era, you would want to take into account that a person might have switched from one drug to another in a class, but that they were still taking that class. So you might actually want your drug era to be across class. But it seems that if you have all of the code for loading the database, that one could modify that fairly easily to do the eras, to construct the eras by therapeutic class instead of by individual drugs, and it would just be a different set of errors. Does that make sense? Mm-hmm.
Yeah, I think where it wouldn't work is in the drug exposure table, because by definition the drug exposure is going to be like a claim, a drug claim or a drug dispensing record. But I think in the drug era that may be allowable. So that's a great question to sort of put out to the rest of the community at some point and find out if anybody disagrees. But I think it could be done. And I don't know about how to handle the switching part, but if you've got a row that indicates someone has a drug era for a specific class that has a start and stop, and then there's another row that's got a different class for that individual with a different start and stop, then you could develop a query that would pull back that sequence, I think.
Yeah, currently drug error is based on ingredient level. It's not even on specific drug. So it combines ingredients of different drugs if they're the same into a contiguous error.
But do you think there's any reason why you couldn't do it the drug class concept ID there? Is that because it's because RX norm doesn't have drug classes and it expects an RX norm or concept ID that maps to RX norm clinical drug or ingredient? I don't know.
Okay, in principle, it can be done. There is no limitation of that. But to categorize, if we have, I didn't look at the categories of the drugs, but there's no limitation to do something like that. But we have to check if the categories exist to categorize them properly.
Right, and then your problem is that there's different ways of categorizing drugs based on the symptoms.
This is Yue Qian. I also want to comment on that. Even though that there are concept ID for drug class, but because in the standard vocabulary that I have found out, there are many coding systems, and by different coding system, you could have your own definition of drug class, and also for individual cases, you may need to customize the drug class definition. So
right.
Even though in the drug era data there is the concept ID for certain drug classes, but I don't think it's possible to meet all the needs.
But if you could define your drug classes and just maybe even dynamically, based on your drug classes, construct the drug error table, or use the code that constructs the drug error table, but substitute in instead whatever your drug class of choice would be, it seems like that might get you there a little quicker if you could borrow the drug error code and modify that to work with whatever drug class system you wanted to work with.
Okay, this is Lu Pinzao. If I can actually just simply kind of deviate it, get to the high-level question that to help me. You talk about is this the OMAP common data model, and I understand there are several common data models. Sentinel probably has its own. And would you be able to comment a little bit in terms of how much of the common data model overlap between Sentinel and the OMAP, and specifically on vocabulary, how much of the overlap do you have? Have you done the assessment? Because that could have implication down the road for your data interpretations.
It's probably easier for me because so within IMEDS lab we do have data, the same data for Truven in both mini-sentinel or sentinel model as well as OMAP common data model. So those kind of data sets are available for people to run experiments in both data models. But the models are different. So OMAP is using common vocabulary that combines information from different diagnoses and drugs into OMAP concepts, where mini-sentinel or sentinel model retains original definitions of the drugs, conditions, and procedures there. So that's one of the differences. Also, OMAP data model has some components that do not exist in mini-sentinel and sentinel model, like cost information. So if you do some statistical analysis of the associated with the cost of the drugs or procedures, this is becoming available within OMAP, and there is no corresponding information within mini-sentinel. In general, if you do just, there's a number of studies that are being done or continue to be done, trying to identify which one is better. I don't see its preference more or less than anything else at this point.
I see. Now, I understand that the NIH said in the collaborative also kind of adopted some of the common data model. I mean, what I'm more worried, I'm not sure in the community people have any convergent opinion about common data model. If we have lots of different common data model, probably defeating the purpose of having a common data model. So I'm not sure any convergent opinion in the community.
there was a lot of discussion at some point about converging there, but I'm not sure if there's work being done to combine them into a single one.
Okay.
Someone else might.
It's important both for FDA down the road, I guess. Then when people actually publish the result and so on, and using different data model, it may actually mean different things. So, okay, thank you.
So this is Susan Gruber. At IMEDS last year, we started a project that's being led by Mark Weiner, and along with Peter MB and I'm blanking on the third name. Anyway, to compare the common data models. And he's got a draft that's just about ready to be submitted somewhere, and then we would end up posting also on the IMEDS website. And his basic conclusion is that from the bulk of the sort of studies one would do, the same information is available formatted in the mini-sentinel or the OMAP common data models, and you would get the same results. The common data model themselves would not be biasing the result. Like Mark said, if OMAP has cost information and mini-sentinel doesn't, then obviously you could only do certain kinds of study in one rather than the other. So that I think will be reassuring when he gets that out. And then there's also some comparison of some specific features. Beyond that, I think these common data models are evolving, and I don't know if they'll converge, but if genomics work becomes important in a certain field, then I think a common data model that includes genomic information will be valued. In another field, that would just be noise to be gotten rid of. If you're looking at health economic data, you might not be looking at a genetic level or a genetic component. So I guess that's my 2 cents. Thank you.
Thank you.
Great. So it looks like we've got 2 or 3 minutes left. Does anybody else have anything they'd like to bring up to Rima or to the group?
Yeah, and I want to invite you, even if you have some conversations that you have offline. My email address is listed in the first slide. So, as we mentioned, we will post our questions to the IMEDS listserv and see what we get as well.
Great. Great. Well, thank you so much, Rima, for joining us. Thank you to everybody else on the phone. So our next call will be Friday, July 17th, and we'll send around an invitation with the topic shortly. If anybody else has any questions about access to the lab or wants to find out more information about the Google group, which is the troubleshooting group we were talking about, feel free to reach out to me, and I'll make sure you get that information. And thank you so much. Enjoy the rest of your day.
Thank you.
Thank you.